so I want to tell you about isoleucine, but first, can you smell maple syrup? I hope it's not coming from a baby's diaper, because that could indicate Maple Syrup Urine Disease, aka Branched Chain Amino Aciduria. So, what the heck am I talking about? So, let me step back. Isoleucine is an amino acid, and it's the fifth amino acid we're going to talk about in our 20 days of amino acids. So amino acids are protein letters. They have a generic backbone with the carboxyl group and an amino group that allow them to link together into chains. You link a bunch of them into a chain, and you get that folds up into form a protein. And how it folds is going to be dictated in large part by the unique parts of the amino acid. So there are 20 different ones that we find in our proteins, and these have these unique side chains, or R groups. And so yesterday we looked at leucine, where we kind of had it was kind of like a snake's tongue. It had the chain with the branch at the end. Here we have the isoleucine, and here, instead of having our branch point at the end, we have it up one. So we have something that looks like this. So this has the consequence of this is you're adding the bulk closer, pushing the bulk closer to the backbone, similar to what we saw with valine. So with valine, we wouldn't have this end group here. So this was valine, and then so isoleucine's like this, and then leucine, it had the branch at the ends. And so this is what we call a sec-butyl group. So yesterday we were talking about, we had an isobutyl group, but sec-butyl group, it's on the second carbon. You have this branch point. But all three of these, so valine, leucine, and isoleucine, are all classified as branched chain amino acids because they have this branching hydrocarbon chain. And because this chain is hydrocarbon, basically this makes it hydrophobic. So water doesn't want to hang out with it. Water excludes it, and they all get, these parts tend to cluster in the center of proteins. So in addition to having those things in common, what these three have in common, these branched chain amino acids, is that they go through similar breakdown processes. They use the same enzyme complexes, so the same protein helper machinery stuff that's going to help break them down. And so if you have problems with this machinery, then that can cause the buildup of intermediates of these amino acids and the amino acids themselves, and then this can cause problems because of things like the amino acids and the products are competing for other amino acids that need to use the same transporters and that sort of thing. This genetic disorders involving mutations in these pathways can lead to things like developmental problems, and can be fatal if not detected and treated early. And so today I want to tell you about isoleucine and this whole idea of MSUD, and how it's and not just the disease form, not just how this breakdown can cause disease, but how this breakdown is supposed to happen, because it uses some really cool enzyme biochemistry. And oh yeah, so as for the maple syrup, that's this side product called Sotolone, and so it's not something that your body normally makes, but it can be a sign of this disease, because it can happen when you have this buildup of all these intermediates. And another thing that can happen if you have a lot of this buildup is, so isoleucine, unlike all of the amino acids except for threonine, it has two stereocenters. So all of the amino acids have a single one stereocenter at the central carbon. So basically, because that carbon is attached to four different things, you can have them attached sticking out in different directions, and those are called different stereoisomers. In isoleucine, this carbon as well has four different groups sticking off of it, so these can stick off in different ways, and then this gives you a second stereocenter. So there's one that we just call isoleucine, and then we have allo-isoleucine, where basically this is swapped. So this would be this would be attached on the back. And so you see, I can't just turn this around, because then it would then this is on a different side. So I would actually have to remove these groups and attach them. Whereas with lysine, where it had it at the end, I could just kind of like swivel it around and it wouldn't be changed. So one of the products that you could test for, like these MSUD disorders, is the buildup of allo-isoleucine, so basically the switch form, which can form when there is like an excess, when there's like a buildup of this, and so that could be used to help detect this disease.
Okay, so we are at day 5 and we are on isoleucine. And so isoleucine, its 3-letter code is ILE, and its initials are I. So we're still in the amino acids where their names make sense and they're easy, but later we'll see that some of them have weird names because the other amino acids already took the names that would make more sense. So remember, so leucine, it has this like snake's tongue, and isoleucine has the branch closer to the center. So because this branch is closer to the center, it's going to make it really unflexible. And so all the peptide bonds, as we've talked about, like, so these are like these planar, you kind of have this chain of planes because you can't rotate around the peptide bond. So the bond linking the amino acids is resonance stabilized. So basically there's double bond character throughout here and you can't rotate around a double bond. So you can't rotate around here, but you can rotate on the sides, but how far you can rotate is going to be dictated by like the bulkiness of the side chain. And so if you have a really bulky side chain, it's going to be hard to rotate. And so you're not going to find it in like alpha helices and that sort of things where you need to be able to kind of rotate more. So isoleucine is sterically hindered. So sterics is where things like basically clash. So it's big and bulky. So it's the same size as leucine, but they're structural isomers. And so they have the same numbers, the same atoms, same numbers of carbons and hydrogens, they're just hooked up differently. So these are different than stereoisomers. So stereoisomers are where they're like hooked up, like they're just like facing, like they're hooked up in like facing different orientations in space. Here they're actually like the linkages are different, not just the spatial positions. So we call these structural isomers. And so leucine, isoleucine, and valine are the branched chain amino acids because they have that branching hydrocarbon chain. And so hydrocarbons, we can basically, if you see, so hydrocarbons form like the skeletal backbone of organic molecules. And basically carbon, it has to form 4 bonds. So it's gonna be like each line represents a single bond. So, and then you can have like double bonds. So a single bond sharing a pair of electrons and a double bond sharing 2 pairs of electrons. If you are confused about all of this, go view my past posts because I don't want to have to go into it right now, or this video is going to get really long and people will get bored. But basically, so these are, carbon has to form like 4 bonds, and so it's or like has to share like 4 pairs of electrons. So it can form like 4 single bonds or 2 single bonds and a double bond. But if you don't have something, basically just like a hydrogen, so that carbon has 4 things. So basically hydrogen, I kind of think of it as like filler for hydrocarbons. And so sometimes if we draw things in like the stick mode, you just like every time there's a point, that's a carbon attached to as many hydrogens as you need to get carbon to be fulfilled its normal state. And then you can have these like different functional groups, and those where you basically replace hydrogen with something more reactive or exciting. But with these amino acids, so with valine, leucine, and isoleucine, so there's branched ones as well as alanine and stuff. These are aliphatic, so they have these hydrocarbon chains that aren't in rings, and these are nonpolar. And so because they're nonpolar, basically this means that, so remember that atoms are sharing pairs of electrons to form these bonds. And basically, because they might not share fairly, and the electrons are negatively charged, depending on if they depending on if they're not sharing fairly, then one of the atoms can end up with having like a partial charge. And so this is what happens with like oxygen and water, and hydrogen and water. So oxygen pulls on those electrons, and so it's really electronegative. And so this makes the oxygen partly negative and the hydrogen's partly positive. You have these partial charges that then attract one another. And so water likes to hang out with itself. But when you have something like when you have a hydrocarbon, hydrogen and carbon share fairly, and so you don't have that polarity, so you don't have partial charges for water to hang out with. Therefore, water will exclude those things, and we call them hydrophobic. So water is basically going to force those things together in this hydrophobic effect. And so if we look at our chart where the protein side chains are ranked by their hydrophobicity, you can see that isoleucine is basically like the most hydrophobic amino acid. And so it really doesn't want to be on the surface, and you'll find it typically in the center of proteins. So yeah, so there's more that are hydrophobic than just these branched chain ones. But the branched chain ones have other special things in common. And so the three branched chain amino acids, remember, are leucine, valine, and isoleucine. And here's what they look like. They're kind of these like white crystalline solids. But typically we're not going to be looking at them like that. We're going to be looking at their molecular forms. And when we're encountering them in our life, it's typically just like through our food and we're not actually seeing it. But if you were to go and look into it, it would look kind of like that. But anyway, so in addition to sharing the same branch structuredness, these branched chain amino acids are going to use some of the same machinery to do their initial breakdown. So first, they're going to get deaminated through this transamination. So with this help of this branched chain aminotransferase. Then this is going to take the amine group and put it onto alpha ketoglutarate to give you glutamate. And then that glutamate can then be, the amine group can then be like taken through the urea cycle to get rid of the amino group, but you can use the glutamate for things, various options. But you now will get rid of this amino group, and this will give you an alpha keto acid. We'll look at more in detail in this all in a minute. And then this is going to go through the second, this complex that they share in common, this branched chain alpha keto acid dehydrogenase complex. So remember, we now have an alpha keto acid, and now we're going to dehydrogenate it. When you hear dehydrogenate, think of oxidation. So this is going to be an oxidative decarboxylation. So more on this in a minute, don't worry. But basically this is going to give you, it's going to remove this carboxyl part, and it's going to attach your alpha keto acid to a coenzyme A. And this coenzyme A, this conjugation with the coenzyme A, then you're going to get broken up further, and you end up with products that can then be used to get energy or to make products. And so we'll see that based on what you start with, you're going to get different end products. So with leucine and isoleucine, you're getting products that can be used for ketogenesis, so you can make ketone bodies and lipids like we talked about in yesterday's post. And then with isoleucine and valine, you're getting succinyl-CoA, which can enter the TCA cycle and be used for glucogenesis. So valine is going to be purely glucogenic, isoleucine is glucogenic and ketogenic, and leucine is ketogenic only. But these classifications are really just kind of yeah, I mean, because everything kind of like interconverts in your cells and stuff. So it's hard to think about them as just strictly one thing or another. But that's basically in terms of the terminology, how it's referred to. So let's look at what's happening with isoleucine. So isoleucine, so first we're going to have that aminotransferase step. We're going to be passing this amino group on to onto alpha ketoglutarate. This is going to give us glutamate. And here we have alpha keta beta methylvaleric acid, EEC. Sometimes abbreviated KMV, thank goodness. So basically, this is a ketoacid. So a ketone is where you have a carbon with a carbonyl carbon, so a carbon double bonded to oxygen with carbons on either side. So this is a common functional group. So functional group is where you basically, remember we have like the hydrogens are kind of like the filler space, and if you replace those with something more fun, you can get molecules that are like more reactive and stuff. And so with a ketone, we have this carbonyl, so this C double bond O attached to a carbon on either side. If we have the peptides, when we have a peptide, that's an amide. So you see this carbonyl a ton of places. And what's really cool about it is that the oxygen, since it's electronegative, it's going to be pulling electrons away from this carbon. And this is going to make the carbon like partly positive and electrophilic, and nucleophiles can then attack it. So yeah, it's cool. But basically, yeah, so you have this, if you have a nitrogen there, we have this amide. Well, also, if you have an oxygen with, if you have this next to like an oxygen, you have, so at the end, like you have a carboxylic acid or a carboxylate when it's deprotonated. And if you have a carbon here, you have an ester. We'll also see in this case later, we're going to see a thioester, which basically instead of an oxygen here, you're going to have a sulfur. This is going to be how we're attaching to the CoA through a thioester linkage, which is going to be reactive and allow you to kind of like swap things off on the other side of the linkage. But going back to our story, so this is going to be an alpha keto acid. So remember, ketone, you have this carbonyl carbon and carbons on either side. And then a carboxylic acid, you have this carbonyl carbon and then this OH or O- once deprotonated. And it's an alpha one because what you have is this, this is the alpha carbon. So this is the same carbon that was attached to the amino group and the side chain and stuff. And so this is the alpha carbon, and so this is alpha keto acid. And so you'll get different alpha keto acids depending on which amino acid you start with. So for example, for valine, we're going to get alpha keto isovaleric acid. For leucine, alpha keto isocaproic acid. But today we're going to focus on isoleucine, so we're going to stick with this KMB thing. But because they're all going through that same first step, it's all like a similar type of thing. They just have different side chains to start with, so you're going to get different products at this point. So next, they're all going to go through this branched chain alpha keto acid dehydrogenase complex. So it's going to decarboxylate it. So it's going to remove this like carboxyl group. And it's going to do this in an oxidative way, which is why this is like a dehydrogenase complex. And we'll look more at this and what this means in a second, and it's going to swap it on to a coenzyme A. Okay, so oxidation. So oxidation and reduction. So much more on this in my post from a couple days ago. But basically, oxidation is loss of electrons or electron density. And reduction is gain of electrons or gain of electron density. And so in biochemistry, oxidation is often associated with like the gain of a bond to oxygen. And reduction is the gain of a bond to hydrogen or the loss of a bond to oxygen. And so the reason why this is basically because when we talk about oxidation and reduction in biochemistry, we're not seeing like full electrons get transferred, like you might see with like some metals and that sort of thing if you're doing like general chemistry. Instead, what we're kind of seeing is like a shifting of the shared electrons. So when they're sharing those electrons in their bonds, because these electrons are in these like clouds that can kind of like, they can hang out more around certain members. And so when you have the when you have something bond to oxygen, because oxygen is so electronegative, it's going to pull the shared electrons towards it. Therefore, the thing that it's attached to when it's oxidized, the thing that it's attached to is the thing getting oxidized, it's going to be losing electrons to that oxygen. So it's not losing like full electrons, but the electrons that they're sharing are kind of getting pulled away. And when you're bound to a hydrogen, so a hydrogen is really bad at, like, it's really bad at holding on to its electrons, so it can easily be like swayed or whatever. And so that's kind of like you're gaining electrons because the hydrogen's really bad at hogging them. So when we're talking about a dehydrogenation, we're seeing here is we're going to be having an oxidation reaction. And so we are going to be losing electrons, and the electrons we lose, we're actually going to give to NAD plus, and that's going to make NADH. And then that NADH can then enter the electron transport chain and be used for making energy, in addition to the bigger payout that you can get if you take the end products of these pathways and then put them through the various, so you can use those electron transfers can then go through this electron transfer chain and give you energy. But the bigger payoff is also going to come when you take those products that you ended up with and put them through like the TCA, the tricarboxylic acid cycle, something's called Krebs cycle or citric acid cycle. And so you can see that isoleucine it can be used to make this acetyl-CoA, which can also be used to make ketone bodies and lipids. And it can also make succinyl-CoA, which can then be used to make oxaloacetate and make glucose. So those are for like making things from them, kind of, except the ketone bodies are often, these can be used as an energy source as well. But going through, you can also just like go through this pathway to break them down further, like because it's oxaloacetate, it's because it's a cycle, it can keep going down and breaking things down and down and down. And so getting as maximum energy you can out of them. So basically, but you can't break down the amino acid, like you can't put an amino acid through this or through oxidative phosphorylation. First, you have to get rid of the amino group and do some like rearrangements and stuff. And so that's what this branched chain amino acid stuff is all about. So what happens with the isoleucine? So when we do this branched chain dehydrogenase complex, what you're going to get up with is this thing attached to coenzyme A. And so in this case, we get alpha methylbutyryl coenzyme A. And so what is this coenzyme A deal all about? So coenzyme A is this common group that you'll see in biochemistry. It's derived from vitamin B5 or pantothenate, and it serves as an acyl group carrier. So an acyl group is where you have a carbonyl attached to some alkyl group. And so an alkyl group is just like a hydrocarbon group. And so this can be like a long chain in the case of like fatty acids. Fatty acids can get attached to CoA, and you have these acyl CoAs, these long, they can be really long ones, and then they can break down when you're doing like catabolizing lipids. What you end up with eventually when you're catabolizing lipids is like acetyl-CoA, which is the simplest form. So an acetyl group is basically, it's just a methyl group attached to a carbonyl. And so this is the shortest that R group could be. And so you have this acetyl group, and when it's attached to this CoA, you have acetyl-CoA. This attachment is done through a thioester linkage. So through one of those linkages, one of those functional groups where we have a carbonyl next to a sulfur and next to a carbon on the other side. And this thioester is reactive. And so you can get basically like you can think about swapping this CoA onto and off of things and swapping this whatever it was attached to onto and off of things. So it's really helpful for being able to carry things around. And so sometimes because it's through this thioester linkage, you'll see this as like S-CoA, but it's like the same thing. This is just emphasizing the linkage form. So acetyl-CoA, so we see an acetyl-CoA because it's what's going to be going like into this pathway. But there are other, I mean, like there are also just like if you have an acyl-CoA, so that can just be like any alkyl group out here. And so what's going to happen when we have this, so this is an acyl-CoA. And so this group is an acyl group on this CoA because you have this carbonyl carbon attached to this alkyl group. And so when we talk about how this enzyme complex is functioning, we're transferring the acyl group from, we're going to transfer the acyl group from the isoleucines and from the alpha keto acid onto this coenzyme A. We can think of it that way as opposed to thinking of just, we can think of it that way as this transfer and the decarboxylation. You're losing that group and you're getting this acyl CoA. And then you have more steps to break them down. But how this acyl CoA is transferred is actually pretty cool because first it's transferred directly onto this enzyme and then it's passed off to coenzyme A. And so as you might expect, this involves a lot of complex molecular orchestration. So speaking of complex, this is done with this branched chain alpha keto acid dehydrogenase complex. And so this complex is kind of like a bunch of proteins kind of loosely held together in this mitochondrial membrane. And so the mitochondria is like the powerhouse of your cell. It's where all of this energy making is going to be happening. And in the mitochondrial membrane is where this can take place. And so we often think, so this is an enzyme complex. And so basically, remember, enzymes are going to speed up a reaction, well, without being used up in the process. And this enzyme complex is actually going to use several helpers or like cofactors. So I like to think of cofactors as kind of like enzyme superhero sidekicks. So these can be like small molecules, like vitamins. So it's like small organic molecules like vitamins. They can be metals. And so the ones we're going to see, the coenzymes we're going to see are going to be like vitamin based largely, and together they kind of like add extra functionality to the enzyme. So the amino acids give it a lot of functionality, having different amino acids, but sometimes you want a little more help, for example, to help you be able to like give and take electrons and that sort of thing. And that's where having these coenzymes can really be helpful. So in this case, this branched chain complex is going to use several cofactors. So, and it also has several complexes. And so when we think about proteins, we typically think about like individual proteins being individual chains of amino acids, but you can also have like multimers where you have multiple protein chains working together to function. And so we call this like oligomers or multimers, depending on like how many there are and stuff. And these can be copies of the same chain, and so they'd be made from the same like protein instructions. And so those would be like homomeric, but they can also be chains from different genes, so like heteromeric. And so what we're going to see is that in the case of this first enzyme E1, this branched chain alpha keto acid decarboxylase, it's going to be a heterotetramer. So basically 4 chains, and they're not all the same. It's going to have 2 copies of this E1A, this E1 alpha, and 2 copies of E1 beta subunits. And so it's going to catalyze the oxidative decarboxylation using a thiamine cofactor. But it's when you have that decarboxylation, you actually get the alpha keto acid is going to be transferred, like it's going to be temporarily held by the enzyme, and then it's going to be passed off to the acyl CoA. So then it's going to be passed off to the coenzyme A, and that transfer is going to be catalyzed by this E2 which is the dihydrolipoyl transacylase. EGADs. Okay, so, and this is also going to help give it the structure. And so it's going to catalyze the transfer of the acyl group from the alpha ketoacids to coenzyme A through a covalently bound lipoyl group, which is like this fatty chain thing. So this lipoyl group, so here is shown lipoic acid, but in the enzyme, it's going to be like covalently linked, so actually like permanently held in there. And it has this silyl groups. And so in this case, this is the oxidized form, but then you can also reduce it and you can attach this to things. And so remember how we saw that there was like that link through the sulfur to CoA? Well, you can also have a link to this sulfur. And so you can have the acyl group, so our alpha keto acid, once we get rid of the acid part and we have this acyl thing, we can add to this, and then this can pass it off to coenzyme A. And so you have this kind of like relay where you're passing off that acyl group. And then you have to reset the system. So you have this E3 group, this dihydrolipoyl dehydrogenase, and it's going to reoxidize that lipoyl group. And so get it back to that SS state where it can then be attacked to have the acyl group stuck onto it, and then it can transfer that to the coenzyme A. So an important note about E3 is that it's shared by other alpha keto acid dehydrogenase complexes. So it's also used by the pyruvate dehydrogenase complex and the alpha ketoglutarate dehydrogenase complex. And so this, because those also need to have some like dehydrogenations happening. And so this E3 is, since it's shared between all of them, if you had a mutation in E3, then that could affect more than just the branched chain amino acids metabolism. It's also important to note that this is like this decarboxylation, this oxidative decarboxylation is basically irreversible. So it's like a committed step in the breakdown. And so you want to make sure that you really do want to break down those amino acids before you enter this pathway. And so you want this to be tightly regulated. And this complex is then regulated by this by these regulatory enzymes, these regulatory subunits. So BCKDK is a kinase, and it is going to add a phosphate group to E1, and this is going to inactivate it. So we often think about kinases as activating things, but they can also inactivate things. And in this case, BCKDK is going to inactivate E1. But once it's inactivated, well, you're going to want to activate it at some point, right? And so that's where PPM1K comes in. This is actually a phosphatase, even though it has a K in its name, it's a phosphatase. And so this is a phosphatase. It's going to remove the phosphorylation that BCKDK put on, and this is going to reactivate it so that you can then break down amino acids again. And so you can have mutations in any one of these subunits, and that could cause this branched chain ketoaciduria. There are different like classifications of MSUD based on like where the mutations are. And then like the severity and stuff is going to depend on part on that and a part on like how severe the mutations are and that sort of thing. So there's lots more you can learn about it, and I'll paste links to like a medical biochemistry page, which is really helpful if you want to learn more. And so the big problem with having, not being able to break down these amino acids isn't just that you can't break down the amino acids for their energy and stuff. I mean, that'd be bad enough. But actually what's happening is you're going to have the buildup of those intermediate products, and that buildup of those products is going to be what's going to be causing a bunch of problems, because those products have to compete with other things that should be happening. And the products themselves can also have like they can like inhibit various ion pumps and that sort of thing to cause problems with energy balance and like swelling and all sorts of different things. But one of the main ways that they can cause problems is that they have to compete with other amino acids to get, like, for the same transporters to get into cells. So these large neutral amino acids, they're like competing for transporters into like really important cells, like in your brain. And if you have a buildup of these products, because you can't, because they can't be broken down, so you get a buildup of them, what's going to happen is they're going to like kind of like outcompete the other stuff that needs to get in, especially because leucine is like, especially because it has a really high affinity for those transporters. So basically, even if there's not that much leucine, because it's so good at like using them, it's going to like outcompete even if you don't have a ton of it. So when you do have a ton of it, then it's going to be a real problem. And so then you can get not enough of these other things that need to get in. And some of the other things that needs to get in are like super important, like tryptophan and tyrosine. Well, those are like precursors for making like neurotransmitters and hormones. So tryptophan, as we'll see later when we talk about tryptophan, it's a precursor for serotonin and melatonin. And when you have phenylalanine and tyrosine, these make dopamine, noradrenaline, adrenaline. And so if you have those having trouble getting in, well, that can be a problem. So those are like the big problems that can come from this. But then there's also like that weird side product, Sotolone, which is going to give the maple syrup smell. And I tried to look into like how it forms or whatever, but it just says it's like a spontaneous product. So it forms spontaneously. So I think if you have a bunch of buildup of those various intermediates, then they can spontaneously form. What happen if you have a buildup of these products is that you can get some of this like allo-isoleucine forming. And so that's, remember where we had that at that second Stereocenter, we had it linked up differently. And so this is, I found the site where you can actually view the different ones. And so the enantiomers, that's where you have, like non-superimposable mirror images. So they're mirror images, like your left and your right hand. Where you have a diastereomer, this can happen if you have multiple chiral centers. And since we have two chiral centers, then if like one is one way and the other is the other way, then you're not going to have a mirror image. And so we call these diastereomers when they're non-superimposable stereoisomers. And so the enantiomers are like the D and the L forms when we talk about amino acids. And then the diastereomers, so here you have like isoleucine versus allo-isoleucine. And so you can look at the various things here. I'll post the link to that.
Oh, and like the other branched chain amino acids, it is essential in the dietary sense, which means that your bodies can't make it yourself. They need to get it through, you need to get it through your diet. These amino acids, in terms of where we find them in our body, they're in a lot of like skeletal muscle. And this is also, they have a bunch of mitochondria there. And the mitochondria are where this BCKD stuff is going down. So it's like a match made in physiological heaven, and you can have lots of this breakdown be occurring in your skeletal muscles, and then that can be used to make energy and all sorts of cool stuff.
As for the nothing really super exciting to say about the discovery of this one. It was discovered by Ehrlich in 1903, and through a lot of careful chemistry, he correctly characterized its structural composition a few years later. And it's coded for by AUU, AUC, and AUA. And it has the same molar mass as leucine, but they're linked up differently because they are structural isomers. So the same number of carbons and hydrogens and all of that, but they're linked up differently. And we're not, when we're talking about linked up differently, we're like at different places, like you have different branch points and stuff. We're not just talking about like they're sticking out different ways in 3D space. So yeah, so I hope this helped you understand isoleucine and appreciate having non-maple syrupy smelling diapers.